So we're going to continue talking about urine formation. And so let's review just a little bit. Remember we said that the kidney has about 1 million of these tiny little structures, which are called nephrons. And remember, the nephron has both a vascular portion, it's got blood vessels, and it has this big long tube portion, the tubular portion, and then it has a regulatory portion. So if you remember when we talked about here, we said that this is the afferent arteriole, and then here's the glomerular capillaries, and then there's the efferent arteriole, and then remember we're going to have another set of capillaries over here, the peritubular capillaries. And also we're going to have the vasa recta. And then we said that if you look at the tubular portion, it starts here. Remember, that's either called Bowman's capsule or the glomerular capsule. And then we have the proximal convoluted tubule, the loop of Henle, and then the distal convoluted tubule, and then finally the collecting duct. And then finally, another thing is that we have a regulatory portion. And the regulatory portion, remember, and I can't show you exactly where it is, but it's where the afferent arteriole touches the distal convoluted tubule. Now in this picture, they don't touch, but they really do. And remember, this is called the juxtaglomerular apparatus. And it has two types of cells. We had the macula densa cells, and then we also had the JG cells. So that's the anatomy of that. And then last time we spent a bunch of time talking about what happens with filtration. So remember, filtration happens right here. And so like I said last time, in order to do filtration, you need three things. You need a liquid, that's blood. You need a force, that's blood pressure. And then you also need a filter. And so we talked about this filter, and this filter, remember, is not a one-stage filter, but it's a three-stage filter. And the first part is in the capillary walls, this part, and then we have the capillary basement membrane, and then finally over here we have podocytes. So it's a three-stage filter. So when you look at it, holes, and the holes get progressively smaller. And remember, what's going to determine what goes through here is the size of the material. So red blood cells are way, way, way too big to go through here. White blood cells are too big. Platelets are too big. Even large proteins are too big. So anything that can go through here, it will. But it has to be smaller than the smallest holes. And we said that whatever goes through here winds up in this tube. And remember, that's called filtrate. And remember, we make about 180 liters of filtrate every day, which means that your entire liquid part of your blood is filtered about 60 times per day. Well, obviously, we don't want to make 180 liters of urine every day. And not only that, remember, the only thing this filter separates things based on is size. So if we look in this filtrate, we're going to see good stuff. Well, if it stays in this tube, it's going to become urine, and it's going to get excreted. So if we want to keep it, we have to get it back out. And that's the second part of urine formation. It's reabsorption, tubular reabsorption. And then sometimes, remember, there are things that we want to get rid of at a higher rate than we can just by filtering it. And so we can actually physically, actively put it in there, and that's tubular secretion. So today, we're going to talk about the last two. We're going to talk about tubular reabsorption and tubular secretion. So So remember, here's sort of a simplified view of our nephron. Here comes our afferent arteriole. Remember, the blood pressure in here is high. And so we're going to have a high pressure pushing against the walls of the filter. And if we figure out this net filtration pressure, remember, it's not 2 like it is everywhere else. It's 10. And because it's 10, lots more fluid is going to come out. Lots more. So it's not 3.5 liters a day. Remember, it's 180 liters a day. But let's look at reabsorption and secretion. So what we've made is we've made filtrate. So filtrate is basically everything that can get through those filter, anything small enough to go through that 3-stage filter. Well, lots of stuff can go through the filter. So things like water, but also lots of ions, all sorts of things. Nutrients, waste products, all sorts of things. And so if you look at this diagram here, this is all the things that go through that filter. Well, if you look at this, a lot of these things are things we would want to keep. Look, we wouldn't want to lose glucose. We wouldn't want to lose amino acids or proteins or vitamins. We wouldn't want to lose those things. We also have to have sodium. We have to have potassium. We have to have these other ions. And certainly we have to have water, because you would dehydrate like crazy if you didn't have water. But there are also some things that get filtered, things that we don't want to keep. Look, urea, uric acid, those are waste products, ammonia, all sorts of things. And so what we're going to do is we're going to reabsorb some things and we're going to secrete some things. So if you look at this picture, if it's blue, it's reabsorbed. So all these things are reabsorbed. If it's red, it's secreted. So all of these things are going to be secreted. And if you look at the picture, it even shows you the part of the tube where it happens. So look, remember, this is our proximal convoluted tubule. So what happens in the proximal convoluted tubule is a whole lot of reabsorption, a little bit of secretion. If you look at what happens in the loop of Henle, we're going to absorb a ton of water and a ton of salt. There isn't any secretion hardly that happens here except a little bit of urea. And then if you look at the distal convoluted tubule, we're going to reabsorb these things. They're basically ions and water, but we're also going to do quite a bit of secretion. So let's look at the individual things. Here's another picture, the same thing as the last one. And if you split up the nephron, you can see what's happening in each one. So again, there's our proximal convoluted tubule. This is what happens. There's the loop of Henle. This is what happens. And then there's the distal convoluted tubule collecting duct. And this is what happens. So let's look at reabsorption first. So reabsorption means that it went through the filter and it's in the tube, but we don't want to excrete it. So we're going to have to get it out of the tube. So in other words, we're going to take needed materials and we're going to move them from the tube back into the blood. And so when we look at that, here's the tube right here. There's the filtrate and here's the blood. So we're going to have to take it out of here and put it into here. Well, it can happen different ways. It can happen by active transport. When we do active transport, we need a carrier, and we also have to use ATP. But because it's active transport, remember, we can move up a gradient. But it can also be passive. Passive means it just happens basically by diffusion or by osmosis. And you do not need a carrier and you don't need energy. So it's going to happen two different ways. Again, it has to go through the tubule into the capillary. So let's look at, and this is a video you can watch if you want. I can't show it to you on here. But let's look at what happens in the first part of the tube. This is the proximal convoluted tubule. So here's Bowman's capsule. And then here's this proximal convoluted tubule. Remember, it's called proximal because it's close to the Bowman's capsule. It's called convoluted, it's twisty. Well, remember the cells here look like this. They have tons and tons and tons of microvilli. And microvilli increase the surface area. They also have tons and tons and tons of mitochondria. Remember, mitochondria are going to make ATP. So we have a lot of surface area, and we have a lot of ATP. And so what happens here is a lot of reabsorption. In fact, this is where most reabsorption happens. It happens in the proximal convoluted tubule. Most reabsorption happens. And so what's going to happen is we're going to pull it out of here and put it back into the blood. Pull it out of here and put it back into the blood. And by the time you make it through here, about 2/3 of all of the sodium and all of the water is reabsorbed. About 2/3 all of the sodium and the water. Well, remember how much we started with was 180 liters. Well, I reabsorbed 2/3 of it, which means that about 120 liters goes back into the blood automatically right here. Now about 2/3 of the sodium is going to go back into the blood. Not only are we going to absorb sodium and water, but we're also going to reabsorb the nutrients. So remember, anything that can go through the filter based on size will. And so there's a lot of nutrients in blood that can go through this filter. Glucose can go through here. Amino acids can go through here. Small proteins can go through here. Fatty acids can go through here. Vitamins can go through here. All kinds of stuff can make it through that filter. And again, if you don't get it out, it's going to be excreted. So we're going to start pulling it out. And basically, by the time we get through here, 100% of all of these nutrients are going to be pulled out of here. So by the time we get down here, there's no glucose left. There's no amino acids, none of that stuff. It's all going to be reabsorbed. We're also going to reabsorb a ton of ions. Now, we already know about sodium, but there are other ions. There's chloride, there's potassium, there's hydrogen ions, there's bicarbonate ions, there's calcium, there's magnesium, there's all kinds of ions. We're also going to reabsorb a lot of those. And then if there are any proteins that made it through the filter, we'll reabsorb those as well. So let's look at that reabsorption. So if you look at sodium reabsorption, it's active, almost always active transport. And so in order to do that, we're going to have a carrier. So here's the carrier, and there's the sodium right there. And we're going to use an ATP. And what it's going to do is it's going to kick it to the other side. So we're going to move it from the filtrate back into blood. And this is active transport. Again, you have to have the carrier, and you have to have the ATP. And because it's active transport, we can move against the gradient. We can move this. So even though there's a lot of sodium already over here, we can keep moving sodium in because it's active transport until there's very little sodium left out here. Let's look at water. Water, remember, follows sodium. It follows sodium. It loves sodium. In fact, for every sodium we reabsorb, we also get 6 waters following it. 6 waters will follow that one sodium. It's not active transport, it's osmosis. So water is reabsorbed by osmosis. It follows the solutes. Let's look at nutrients. So nutrients, remember, they enter the tube. We don't want them to get excreted, and so we're going to have to get them out. Well, let's look at, say, glucose. So if we look at glucose, if you remember, glucose is in your blood. We try to keep it pretty much the same. It ranges anywhere from about 70 to 110 milligrams per deciliter. Ideal is about 90. It does go up and down. But anyway, that means that 90 glucoses is going to come into here. So there's the glucose, 90 of them. Well, as they move through here, there are little carriers all along the walls here that absorb glucose. So here they are. And as the glucose goes by, they grab it and reabsorb it. It's by active transport. So they grab the glucose, grab the glucose. Well, as long as the glucose is below a certain amount, every bit of the glucose is going to get grabbed. And so when I get over here, I'm going to have 0 glucoses. But I don't have an unlimited number of these carriers. I only have a certain number. And so they only can get out up to a certain number of glucoses. And if you get above that number, glucoses are going to escape. That number is called the transport maximum, or the TM value. And I don't know what the TM is, but let's say it's 200. So in other words, I can reabsorb up to 200 glucoses per minute. Make sense? So what if it's 90? That's what a normal person is. 90 is obviously less than 200, which means I'm going to reabsorb all of the glucoses. So when I get over here, there's going to be 0 glucose. What if I just ate a meal, though, and my glucose goes up to 150? Is 150 less than 200? Yes. So how much glucose is going to wind up in my urine over here? How much is going to be excreted? 0. All the glucose is going to be reabsorbed. But what if my glucose level is 201? Is 201 less than 200? It's not. I'm going to absorb up to 200, but what's going to happen to that 1? That 1 is going to escape. And that glucose, wrong color, that glucose is going to make it all the way. And I'm going to have 1 glucose in my urine. Does that make sense? What if it's not 201? What if it's 215? What does that mean? How many are going to wind up in my urine? 15. What if it's not 215? What if it's 1200? How many wind up in my urine? 1,000. That's right. That's exactly right. So look, when you're a diabetic, the problem is not at your kidney. The problem is your blood glucose levels are higher than your transport maximum. So if you do have diabetes mellitus and you're not controlling it very well, your blood glucose probably won't be 90. It might be 120. That's okay. It might be 150. Now you're starting to get up there, but it could be as high as 1,200 or 2,000 or even higher. But every single glucose above the transport maximum is going to wind up in your urine. So you're going to have glucose in the urine. Well, remember, water follows solutes, and those glucoses are solutes. And look what happens. And when this glucose gets down here, it sucks water back into the tube. So what happens to urine volume? It goes way up. Remember, one of the cardinal signs of diabetes mellitus was polyuria. And remember, if you pee a lot, you're also going to be very thirsty. And so you're going to have polydipsia. And think about it. What's going out? Not just water, but glucose. Glucose, which is our primary energy source. It's what we're going to use to make ATP. It's our food source we're going to use to make ATP, but it's being excreted, which means you're not going to get any benefit from it, which means you're going to be very, very hungry. So these people also eat a lot. So these are those 3 cardinal signs, means present in all cases of diabetes mellitus. And it's totally 100% because the glucose is higher than the transport maximum. And so we're going to excrete glucose. Does that make sense to everyone? So if I were to graph that, it would look like this. So look, here's glucose. And so my blood glucose, as it goes up, well, look, I haven't reached my transport maximum yet. And so as long as it's here, as it's 10, 100, 110, 150, as long as it's less than my transport maximum, look how much winds up in the urine, 0. But the second I reach the transport maximum, remember we said, for an example, our TM value is 200. But look what happens when I go to 201. 1 winds up in the urine. Look what happens when I go to 210. 10 winds up in the urine. Look what happens when I go to 250. 50 winds up in the urine. Look, these lines are parallel to each other. In other words, every single one that's higher than my transport maximum, every single one of those is going to be excreted. It's going to wind up in our urine. Does it make sense? Any questions? Well, that's not just true for glucose. It's not just true for glucose. All organic nutrients have a transport maximum. So it could be an amino acid. So amino acids also have a transport maximum. And so I don't know which amino acid we're talking about, but there's a certain level in our blood, whatever it is. Let's say it's 12. Well, that's what's going to get to the kidney. That's going to go, and it's going to go through the filter. Amino acids go through the filter very easily. And so look, there are carriers for that particular amino acid. And I don't know what this transport maximum is, but let's say that transport maximum is 20. So just like glucose, this is less than 20. 12 is less than 20, which means that every single one of those amino acids is going to be reabsorbed. And so if I look over here at the urine, I look for the amino acids, there's going to be 0 of that amino acid in the urine. Well, what happens if amino acid level goes up and it goes to 30? What's going to happen to all those extra above 20? They're going to zoom on by, and I'm going to wind up with how many in my urine? I'm going to wind up with 10 in my urine. Exact same graph, look. Exact same graph, but instead of glucose, it's amino acids. And so there's the transport maximum right there for this amino acid, and we said it's 20. So as long as I'm below 20, none of it is going to wind up in the urine. But the second I go above 20, every single one is going to wind up in the urine. Does it make sense? Any questions? So this is true for glucose. It's true for all the amino acids. And remember, there's 20 different ones of those. It's true for fatty acids. It's true for all organic nutrients, whatever they are. They all have a transport maximum. And if I exceed the transport maximum, I'm going to excrete some of those nutrients. Any questions? Can everybody interpret this graph? And like I said, all you'd have to do is change the words and put whatever it is that you wanted to talk about here. And the graph would look basically exactly the same. Now the TM value is going to be different for every nutrient, but the graph is not going to change at all. Remember, we look at the cells of the proximal convoluted tubule. They look like this. So they have a lot of microvilli. That's going to increase the surface area, make it easy to reabsorb. They have a lot of mitochondria, and that's where we're going to get our ATP, because a lot of this, remember, is active transport. And then if we look at water, remember, water is by osmosis. It follows And then most other ions and fat-soluble substances are by diffusion. In other words, they go down their gradient. They cannot go against their gradient. So here's our filtrate, there's our blood, and we're going to absorb all these different things. So active transport, that's the red lines. And then passive transport, that's the blue lines. Any questions? Okay, some substances are not reabsorbed at all. So they get in the tube and they just stay in the tube. They're not reabsorbed at all. So if they're too big, they can't go through the membrane. They're not lipid soluble, they can't go through the membrane. Or they don't have a carrier, they can't go through the membrane. And if they can't go through the membrane, they have to stay in the tube. And so they get excreted. And so if you look at some of the things that are like that, they're these, and these are waste products. Those are waste products. And so those waste products then are not reabsorbed, and so they're excreted. Now urea is reabsorbed at a slight rate. This one can be reabsorbed by diffusion, but not that much. Remember, it can only go down its gradient. So that's what happens with reabsorption. Any questions? Okay, so we went through the proximal convoluted tubule, but look, we're not done. We're going to go into the loop of Henle. Remember, it looks like this. It has two limbs to it. And don't forget, we have two different types of nephrons. Remember, we have those that are short. They're called cortical nephrons. And if you look at their loop of Henle, it's only like this long. It's not a very long loop of Henle. But we also have those juxtamedullary nephrons. And they are much longer. They hang down like this. Well, cortical nephrons are almost completely enclosed by the cortex, but juxtamedullary nephrons hang down into the medulla. Well, let's look at the cortex versus the medulla. There's salt in here. All of our body fluids are salty. And if you want to measure how much salt, how much solute is in our body fluids, we use this term. It's called osmolality. So osmolality is just how salty, how much solute we have in our fluid. And so we count how many solute particles there are in 1 kilogram of water. So if I look at this, that's 1 kilogram of water. If I look at this, this is 1 kilogram of water. I count how many there are. And if the more of these you have, the higher the osmolality. So when you look at this one, it's got a lot more solute than the first one did. And so this one has a low osmolality. This one has a high osmolality. And osmolality can cause osmosis. So you may have done this experiment back in general biology. Do you remember where you took a U-shaped tube like this? And you put a membrane right here? And it was a selectively permeable membrane. And then you put equal amounts of water here and here. but I'm going to add some solute to this side. And the solute is too big to go through the membrane. Now there's a little bit of solute over here, just like these two things right here. Well, the solute can't move. You remember what happened instead? What's going to happen instead? Nobody remembers? Something's going to move. Do you remember what it is? What's going to happen is the water is going to move. Water is going to move. And you may have heard this, but this is the easiest way to understand it, is that solute pulls solvent. Remember, solute is what dissolved, and solvent is the water or whatever it's dissolved in. So look, if I look at these two sides, A and B, which side has more solute, A or B? Which side has more solute? B does. Look, the solute in B is going to pull the solvent. These things are not going to change. But what's going to happen is water is going to move, and it's going to move from A to B. And so what it's going to look like when it's done is this water on this side is going to go down like that. And the water on this side is going to go up like that until the concentration of solute is the same on both sides. Now, that's not the amount. It's the concentration is the same on both sides. We're going to have the same concentration. So in other words, solute pulls solvent. It pulled water this way. It pulled water until they are equal. So we got osmosis. Well, if we look at our body fluids, they have this solute in them. They have this osmolality. And it's the unit is milliosmoles. And you don't really need to know that, but it's just the unit. And if we look at our blood has a certain saltiness, and it's 300 milliosmoles. 300. If we look at our tissues, our tissue fluid, our interstitial fluid, it's 300 milliosmoles. That's how salty we are. But the kidney is the weirdest thing, because the deeper you go into the kidney, the saltier it gets. So it starts at 300, but when you enter the medulla of the kidney, it starts getting saltier and saltier and saltier the deeper you go. So look, it looks like this. So out here, it's normal. It's 300, 300, 300. But when you go into the medulla, the deeper you go, the saltier it gets. Look, it goes 300, 400, 500, 600, all the way to about 1200. 1200 milliosmoles. In other words, it's very, very salty. Well, look at our nephron. Most of the nephron is up here where it's 300. But the loop of Henle and the collecting duct are not. So the loop of Henle hangs down in this salty fluid. And remember, we have two different types of nephrons. We have the ones with the short loops of Henle, and then we have the ones with the long loops of Henle. They both hang down. So right here is the dividing line between the cortex and the medulla. So here's the cortex, here's the medulla. Well, if we look at how salty the fluid is here, it's 300, 300, 300. But remember, when you get down into the medulla, the deeper you go, the saltier it gets. So look, it went down. And the deeper you go, the saltier it gets, all the way to about 1,200 milliosmoles. So if I look at my cortical nephron, it only hangs down to about this number. But if I look at the juxtamedullary one, look, it hangs all the way down here. Well, don't forget that solute pulls solvent. The water got pulled by the salt. So look, this is salt. The deeper you go, the saltier it gets. So look what happens. Water with salt in it is going to come into our tubule. Here it comes. It's going to come in. And right here, it's 300. So look, it's 300 here, 300 there. Is water going to move? Yes or no? It's the same on both sides. Is water going to move? No, that's exactly right. But look what happens when this 300 gets here. Look what happens. Now there's more salt outside than there is on the inside. There's more salt on the outside. Salt pulls water. Water's going to move. Which way is it going to move? Into the tube or out of the tube? It's going to move out until this number is the same as that one out there. It's going to move out until this number is the same. But now look, 400 is going to go down here. What's going to happen now? 400 is going to go down here. Which way is water going to go? It's going to go out until this number is the same. It's going to go out until this number is the same. And if I go down here, more water is going to go out until it's the same. And more water is going to go out until it's the same. And more water is going to go out until it's the same. So when I get down to the bottom, it's going to be about 1,200 milliosmoles. So look, water left the tube. Water left the tube. And it's going to stay in the body. It's not going to be excreted. In other words, I'm going to save water. And so if I look at that, it looks like this. There's that tube hanging down in there. And water is going to leave this tube. So look, here comes my tubular fluid. Remember, it's 300. But water is going to leave. So now it's going to be 400. Water is going to leave. It's going to be 500. And it's going to do that all the way down because remember, solute pulls solvent. And so look what's going to happen. this pulls water until they're the same. It's going to pull water until they're the same. And that's what we're going to see here. So here comes that fluid at 300. Water's going to go out. Water's going to go out. Water's going to go out. Water's going to go out all the way down this tube. And as it does that, as water goes out, this concentration is going to get higher and higher and higher. So again, there's where the cortex is, 300. But it's 400 out here. See, it's 400. Water's going to leave until it's 400. Water's going to leave. Water's going to leave until it's 600. Water's going to leave. Water's going to leave until it's 1200. So water leaves the tube. That water will stay in the body. It's not going to be excreted. Does that make sense to everyone? Any questions? Okay, but look at here. What happens when I turn around and go back this way? Look, this is 1200, but it's only 900 out here. What's going to happen when 1200 gets here and it's 900 on the outside? Water's going to come back in until it's 900. And if I went all the way back up, it would be 300 again back here, and I would have done absolutely nothing because the water would just come back in. And I'd have the same amount of water that went out, it would come back in. But remember, these two sides of this loop of Henle are different. Remember, the descending limb has simple squamous epithelium. But the ascending limb, look, it's thicker. The ascending limb has simple cuboidal epithelium. And the descending limb is permeable to water. But the ascending limb is not permeable to water. It's impermeable to water. And so the water that went out can't get back in. Makes sense? So the water that went out can't come back in. But here's something. Look at this. When in all of your whole science career have you ever seen a situation where there's a lot of salt in one place and very little salt in another? It's in fluid. What's going to happen if I drop salt in a glass of water? What's going to happen to that salt? Or sugar or anything else? What's going to happen to it? Is it going to stay like this? It's going to dissolve. And if I let it sit there very long, if I drink that water, no matter where I get the water from, it's going to be salty water. In other words, diffusion happens. So something weird is going on here because it doesn't do that. This is 300, this is 400, this is 600, and so on. Something has to be maintaining this salt gradient. Otherwise, over time, it would just disappear. Well, what maintains it is that on this side, there's a bunch of carriers. So water comes out on this side. Remember, this is thin over here. This is thick over here. The water can't come back in. But look, there's a bunch of carriers. So look how much salt I have in here. I've got a bunch of salt. So I start taking some out. That number is going to go down. Look, goes down. But I'm going to keep taking the salt out. So this part here is going to be very salty because I'm putting salt out there. But there's less salt to pull out here, so it's going to be a little bit less salty. I'm going to keep pulling out salt. It's going to be even less salty. I'm going to keep pulling out salt. It's going to be even less salty. So it's going to look like, when I look at it, is that down at the bottom, because I have the most salt to pull out, it's going to be the most salty. But as I have less and less to pull out, it's going to be less and less salty. But as I do that, what's going to happen is I'm going to maintain this gradient. And so, again, the deeper you go, the saltier it's going to get. But it's because on this part of the loop of Henle, I'm pulling salt out like crazy. And so if you look at the loop of Henle, on this side, I pull water out. On this side, I pull salt out. And that's what maintains this increasing salt level so that I can keep absorbing the water. And I'm going to pull out so much salt that, look, it's even less than what I started with. So in other words, I'm going to save a bunch of salt. But there's still a little bit in here. And there's still water in here. There's salt and there's water. But look what happens next. It's going to go through here and it's going to go into the collecting duct. And the collecting duct goes right back through this increasing salt concentration again. So, if I look at this, this is permeable to water. Permeable. So, I'm going to automatically save water. The other side over here is impermeable to water. And so the water can't go back in, which means I'm going to save water because it can't go back in. But this over here is variably permeable to water. In other words, I can control it. And so I can make it high or I can make it low. I can save water, or if I don't need it, I can excrete water. So let's look and see what controls this. Remember, it's variably permeable to water. What controls it is a hormone. So, descending limb, permeable to water. Water comes out. Ascending limb, impermeable to water. Water stays in. But I'm going to transport sodium and chloride out. But in the collecting duct here, it's variably permeable to water. And what it depends on are these hormones. And we've gone over these hormones before. But let's review them a little bit. So it's ADH, aldosterone, PTH, and so on. And so if we look at ADH and aldosterone and A and P, they're going to have an effect on water because those are going to have an effect on sodium. PTH is going to have an effect on calcium. But let's look at ADH first. So ADH, remember what this stands for? It stands for Anti-Diuretic Hormone. It's an anti-peeing hormone. So in other words, I don't want that water to go out. I want it to stay in the kidney. I want it to stay in the body. So what ADH does is it makes this permeable to water. So if you are dehydrated, the brain monitors that. And if you're dehydrated, you're going to release ADH. ADH does two things. One thing is it's going to make you thirsty, so you'll drink water. But a thing it's going to do is going to go to the kidney, and it's going to go to the collecting duct. And what it does is at the collecting duct is it makes the collecting duct reabsorb water. So look, what's going to happen is with ADH, this is going to be permeable to water. Well, remember, it goes right back through that increasing salt concentration. And as it does so, water is going to come out. And so remember, the amount of water that's going to come out is going to be the same as what's out here until the solutes are the same. And so I'm going to save water. If I don't have ADH, the water is going to stay in the tube. So look, it's impermeable to water. And so without ADH, impermeable to water, the water is going to stay in the tube. It can't come out, so the water then is going to be excreted. So if I look at the difference between when I do have ADH and when I don't have ADH, again, it's all about this collecting duct. So with ADH, it's permeable to water. Permeable to water. And so the water comes out. So look at all the water coming out. And as the water comes out, the urine is going to get more and more concentrated. So what I'm going to have is a very small amount, a small volume of highly concentrated urine. But if I don't have any ADH, this is impermeable to water. Impermeable. So the water can't cross the membrane. So even though I'm going through here, the water can't get out. And so the water stays in the tube. And so what I'm going to have is a large amount, and it's going to be very dilute. So this is a nice little summary slide of what happens with ADH. Water comes out. What happens without ADH? Water stays in. And so you can see here. And so this is a nice place for you to come and figure out what happens with ADH. So again, if you're dehydrated, don't want to lose any more water. So the brain, the hypothalamus, detects that. You're dehydrated. I need to save water. And if you want to save water, you don't want to make a lot of urine. So it sends a signal. We're going to make the collecting duct permeable to water. And so the water is going to be reabsorbed. And then we're also still dehydrated. And so ADH also makes you thirsty, so that you'll drink water. But again, it makes this permeable to water. So with ADH, permeable. Without ADH, impermeable. Any questions? Here's another summary slide. So again, we're trying to keep the salt concentration as close to normal. Our brain is going to monitor that. If you're dehydrated, this is what's going to happen. If you're overhydrated, this is what's going to happen. Let's look at aldosterone. So remember, aldosterone is a steroid. It comes from the adrenal cortex. And what it does is it makes you save sodium and excrete potassium. So we're going to save sodium and we're going to get rid of potassium. But remember, what follows sodium? Water. So if we're going to save sodium, we're also going to save water. So that's exactly what happens. It comes from the adrenal cortex. We're going to save the sodium, which is going to also save water. And then we save water. It's going to go into the blood, so we're going to increase blood volume. We're going to increase blood pressure. Well, that water had to come from somewhere, so it came out of the urine. So urine volume goes down. And so it happens in the same part of the tube. It happens right here in this collecting duct. And so what's going to happen is we're going to somewhere. We're going to save sodium. That's not really where I wanted to see it. But anyway. Any questions? That's still ADH. I guess I don't have a picture of it. I guess I don't have a picture. But anyway, that's reabsorption. We also have reabsorption of calcium with PTH, and we also have reabsorption of sodium with aldosterone, water with ADH. But let's look at secretion. So secretion is the opposite. It's where we're going to take stuff out of the blood and put it into the tube. And so we're going to get rid of extra, whatever it is. And these are the things that we're going to secrete. So we secrete things like creatinine, ammonia. When we have aldosterone, we're going to do potassium, hydrogen ions. But we're going to take it from the blood and put it into the tube. And so it's going to go in this direction. This direction. Take it from the blood and put it into the tube. Again, it's going to happen by active transport. Here's another little video. You can watch it if you want at some point. But what it's going to do is it's going to get rid of excess stuff. So if we have aldosterone, remember we're going to save sodium, but we're going to get rid of potassium. And so that potassium is going to be excreted. We're going to secrete it into the tube, and it's going to wind up in the urine. Same thing with hydrogen ions. We're going to secrete it into the tube and excrete it into the urine. And then the other hormone that's involved with this is ANP, and we talked about it. So ANP makes you get rid of sodium. So we're going to take sodium and secrete it into the tube and excrete it into the urine. Remember what follows sodium? Water follows sodium. So we'll also excrete water. That water is going to come out of the blood, so we're going to decrease the blood volume. If you decrease the blood volume, automatically you decrease blood pressure. And so we're going to restore homeostasis here. So aldosterone, ADH, I don't see A and P on there. Anyway, so if you summarize what's going on here, remember here we had filtration, we talked about it, and then this is what happens at the proximal convoluted tubule. Look, we're getting a ton of reabsorption. Could be by active transport, could be by passive transport. But remember, as we go through here, we're going to absorb 100% of the organic nutrients, and then about 65% of the sodium and 65% of the water. And then we move down here, we're going to absorb more water. So this side, water. On this side, we're going to absorb sodium and chloride on that side. When we get here, it's variable. It depends on those hormones. And the hormones are ADH, and aldosterone, PTH, and ANP. And so what's going to happen here is this. So we're going to get some active transport, but it's variable. And then finally, when we get over here, we get both secretion and absorption, depending on these hormones. So proximal convoluted tubule, all of that stuff gets reabsorbed. 65% of the volume, that's the water, about 100% of all this stuff, and so on. That's what's in here. And then here's the thin segment of our loop of Henle. We're going to absorb water. It's permeable to water, so water comes out. But on the other side, remember what's going to come out is salt. We're going to take salt. It's impermeable to water, but salt's going to come out. And then we get up to here, it's hormone regulated. PTH, aldosterone, depends on the hormone. And then when we get to the collecting duct, it's also hormone regulated. Any questions? Let's look at urine real quick. So everybody in here is pretty familiar with urine. You see it all the time. So we know what color it has. It's always some sort of yellow color, unless you've eaten something different, because there are certain things that can change the color. Vitamin supplements, foods, drugs, they change the color. But the color of urine, remember, is this sort of yellow color, and it's due to urobilin. So remember, urobilin is the breakdown product, or one of the breakdown products of hemoglobin, and it gives our urine its normal color. Well, depending on the concentration of urobilin, it can be very, very different in terms of how deep the color is. But when you look at a scale, you can see this. And so when you look at this, this is very, very dilute urine. But here it's very, very concentrated urine. And so depending on how hydrated you are, your urine's going to look like this or like this. So if it looks like this, you're very hydrated. If it looks like this, you're very dehydrated, because what's happening is the concentration of the urobilin goes up. You should also probably already know what the odor of urine is it doesn't really have too much of an odor when it's fresh, but if you let it set for a while, it develops an ammonia odor. But you can change the odor of your urine by what you eat. If you eat asparagus, anybody here eat asparagus? If you eat asparagus, it's got something in it called oxalic acid. And you can smell that in the urine. How many of you drink coffee? If you drink a lot of coffee, your urine smells like coffee. Or if you take certain drugs like ampicillin or penicillin or tetracycline or lots of different drugs, the urine smells like those drugs. There are other things that can change the smell of urine. Look, sometimes urine smells like fruit. It smells like maple syrup. It smells like sweaty feet. These are all actually conditions or diseases. And so smelling the urine actually can kind of tell you what's going on in the person's body. So people who have the disease called phenylketonuria, their urine smells like mice. It smells mousy, and so on, all the way down. If you look at the pH of urine, it varies. We can vary it depending on what we need to do. Sometimes we need to get rid of acid, and so we get rid of it in the urine. And so we have an acidic pH. So if you eat a lot of meat, you tend to have an acidic pH. On the other hand, sometimes we get rid of base, and so it's in the pH. And so if you eat a lot of vegetables, your pH is going to be higher. Look at the range. This is the absolute range. You can't get any lower than 4.5, and your urine can't get any higher than 8. So that's the range that we can make. It also has a lot of solutes in it. And so solutes are going to change how thick it is. So the more solutes you have in here, the denser it is. It has a higher what's called specific gravity. So if you look at, say, fresh water versus, say, ocean water, fresh water has a little bit of solutes in it, but ocean water has a ton of solutes in it. That's why it tastes salty and all that. So this one has a much higher specific gravity than this one does. So the more solute that's in the urine, the higher the specific gravity. And then if you look at what's in urine, it's mostly water. But there's a lot of other stuff in urine too. There are waste products, there's solutes, there's all kinds of things in it. And so if you go and you have a pH test on your urine, you can see what that tells you a little bit also. And then finally, from the kidneys, we go into the ureters. Remember, the ureters are just tubes. They're tubes that carry the urine down to the bladder. But when you look at them, they have smooth muscle in them, which means that you can push the urine down. You can push it down, almost like milking. And so even if you're laying flat on your back, you can make urine move to your kidneys. Even if you're hanging upside down, you can make urine move to your kidneys. And then we get to, well, here's what the ureter looks like. So see the smooth muscle? That's what that is. And don't forget we have transitional epithelium right here. We talked about that back in A&P1. Sometimes things that you eat will crystallize. And so you can wind up with kidney stones. Here's what a kidney stone looks like. Look, they're not smooth. They're jagged like crazy. And as they go through this tube, they cut it and tear it and rip it as they go all the way down. So kidney stones, when you pass them, hurt like crazy. In fact, it's probably the closest to having a baby that a man will ever get to. That's how much pain it is. But it's mostly calcium or some other mineral. And then we get down to the urinary bladder. And if you look at the urinary bladder, remember it's a sac for storage, and it can expand. So as it expands, the walls stretch. Remember, we also have transitional epithelium in here as well. And then there's a layer of muscle here, and that's called the detrusor muscle. And so as the bladder fills up with urine, eventually we get a reflex, and the muscle contracts and forces the urine this way. If you look at the bottom part of the urinary bladder, there's a triangular shaped smooth layer right here. And the urine just sort of slides across it down toward the urethra. It's called the trigon. And then remember, we have transitional epithelium. We have this detrusor muscle. There's also connective tissue. It's like a balloon. When it's full, the walls are smooth. But if you take the urine out, the walls inside fold up like an accordion. And these are called rugae, these folds. There you can see some rugae in there. And then we have the urethra. So the urethra is just the tube to the outside. Well, that's the tube right there, but we don't want urine coming out all the time. So there are two rings of muscle that keep the urine from coming out. They're called sphincters. And so sphincters are bands of muscle in a circle. And when they contract, they close off the tube. Well, there are two of them. There's an internal sphincter, which is smooth muscle, and it's involuntary. So here's the internal sphincter right here. But there's also an external sphincter, which is skeletal muscle. And because it's skeletal muscle, it's voluntary. It's where it passes through the abdominal wall. So there's the external sphincter. Well, if you look at this is a female. Females have very short urethras. It's only about 1.5 inches to 2 inches. But if you look at a male, the urethra is much, much longer. With the male, it's about 20 centimeters in length, and it's divided up into 3 regions. So the first region goes through this gland. The gland is called the prostate, so it's called the prostatic urethra. The next region goes through the body wall, and that's called the membranous urethra. And then the last part through the penis, and that's the penile or spongy urethra. So there's 3 parts: prostatic, membranous, and spongy urethra. When we void the bladder, that's called micturition. And what's going to happen is when the walls stretch, it's going to send a signal to the central nervous system. And what's going to happen is we're going to turn around and send the signal back to tell this to contract. Well, remember there are these 2 sphincters. There's one right here and one right here. Well, when the central nervous system tells the bladder to contract, it tells this one to relax. And so the urine then starts getting pushed out. And the only thing keeping it from coming out all the way is this voluntary or external sphincter. And that we have voluntary control over. until the pressure in here exceeds the strength of the sphincter, and then it's going to come out whether you want it to or not. But, so there's two parts to this. There's one part that's involuntary or reflexive, and then there's another part that's voluntary. And it looks something like this. So the control of this happens here at the base of the spinal cord, but also up in the brain. So it looks like this. So there's the involuntary part, and then there's the voluntary part. Any questions? So we pretty much finished this chapter. So if you come back tomorrow, we'll review some of this, and you can ask questions if you want. That's pretty much the end of that chapter. So I'm going to go ahead and stop the recording now.